Okay. Oh, do join us. Okay, thank you, Richard. It's a pleasure to be, well, no, actually, I'm getting flashbacks being back in this room to exam boards and such like exam checking meetings. That's right, so Neil will remember these. Sitting in here deciding about whether you should have a comma after and in a list, I seem to remember, was one of our happy hours spent in here. So it's nice to be here for a different reason than an exam setting meeting. Thank you all for coming. So what I want to talk about today is really quite general about the idea of modeling decision making in populations of cells. I'm particularly interested in populations of cells making decisions during development, where cells in a multiplying population are trying to decide what they're going to do in the adult body plan. But I think the questions are fairly general, and so I've tried to pitch the talk in a reasonably general fashion without going too much into the biology. But obviously there's a lot of biology underlying this as well that I'm not really going to talk about. So the overview, the broad question that I'm interested in is how, when you have populations of cells that initially have equivalent potential to form different differentiated cell types, how is the pattern and the tempo in which they adopt particular fates, how they make particular decisions, controlled? And that's clearly of central importance during development. That's one of the key processes that's going on in there. And I'll probably only talk about two of these because otherwise it will take too long. I'd like to talk about all of them, but I'll probably focus on these two questions. I want to look at situations where this decision has to be made rapidly, and in many developing organisms you want to get your basic body plan set up as quickly as possible because you need to start eating and running away from predators. So if you imagine developing in a pond, for example, you want to get your development done quickly so that you can start that process of eating and evading being eaten yourself. There you need to be quick. But there are other situations during development where you need to generate a very diverse set of cell types. And a classic example of this is in the development of the vertebrate nervous system, so your nervous system, for example, where you want to develop many, many different types of cells over a long period and lots of spatial complexity. And there you have a much more measured and a much more adaptive decision process going on. And so I want to talk about the question of how you balance decision making with not making decisions too quickly. So kind of much more measured and deliberate decision making process. And then, like I say, in the unlikely event I get time, I can talk about a different situation where you need to adopt different strategies for doing this to the ones that I'll talk about here. But this is in plants, whereas these are in animals. But I think it's probably too much. Okay, so let's start with the basic idea of rapid decision making. So this has been studied in many, many different contexts in developmental biology. Probably the earliest of these come from fruit fly, the classic model organism of developmental genetics. And the body of the fly is covered in small bristles that you can't really see very well at this resolution. But if you look at a close-up on say the back of the fly, although these bristles are pretty much everywhere, you see these nicely spaced sensory bristles in here in a nicely arranged pattern. During development, these cells, the cells that prefigure these bristles, the precursors to those bristles, because each bristle has associated neurons and various other cells with it, come from cells that are singled out from the same population of cells that actually forms the skin, the surface layer of the fly. In this process called lateral inhibition, where you're selecting out a subset of cells. And the way that this actually works in the fly, if you look in the embryo when this is developing, this pattern in the adult is being laid down, you actually get these cells are singled out. This is looking at gene expression of one of the genes that's essential for the sensory organ precursor fate that goes on to form the bristle and associated cells. And they actually form in rows. They're not quite beautifully straight rows because this is biology. But if you look slightly earlier, all the cells in a row down here and various other rows, there's a discrete set of rows forming these bristles. Each of these has a initially equivalent potential to form either a bristle precursor or a skin precursor cell, essentially. And what's happening schematically is that we have a population of cells, which in this case is just a line of cells, and by a mechanism called lateral inhibition, you single out a subset of those cells to go on to form the bristle precursors. And so that's the most basic type of decision process you can have. It's a binary fate decision. Do I adopt this pattern of gene expression, which will lead me to a particular fate, or do I adopt a different pattern of gene expression that will lead me on to a different fate? And this can also be, these cells will stop proliferating, whereas these ones will carry on proliferating. You've got more cells in between these bristles than you have bristles. So you need proliferation to carry on in here as well. This can happen in many, many different contexts. Different type of bristles in flies actually form from small clusters of cells rather than lines, and you single out one. And you can show this, that this is an adaptive process in here. A simple mechanism for this would just be that cryptically, these cells are actually predetermined to be bristle precursors. We just can't see it, but that they develop into bristle precursors by a mechanism that we don't currently have markers for. But you can show that that's not necessarily the case in here, because you can actually ablate the cell, can kill off the cell that's going towards this, what's called the primary fate, and then one of these other cells will take its place. They can now adopt the primary fate. They're being inhibited from doing so by this cell. So it is an adaptive process that's a balance between different cell fates in here. It's not just that there's a winner and nobody else has got a chance. And this can also happen in 2D populations of cells. This is a picture of a sensory patch in an inner ear, a vertebrate inner ear. This is from a zebrafish. So these are results from my wife's lab, Tanya Whitfield, in biomedical science. And here, the cells, but this is a surface view, and these cells that you can see actin bundles in, these are sensory bundles, are the cells that are singled out in this type of process. But now it's in a large 2D sheet of cells where you're singling out a whole subset of cells. And that's the same mechanism is underlying all of these different decisions. The same molecular mechanism underlies all of these decision processes. And there are many other examples that we could give of this type of basic binary fate decision making going on during development. And the mechanism that underlies that has been mapped out by a number of decades of initially genetic experiments and then increasingly biochemical, biophysical experiments to actually map out what's going on during those decision-making processes. And it centers around these two types of proteins, delta and notch, that are actually membrane-bound proteins. They're sat in the membranes of cells. And they can interact by direct protein-protein interaction between immediately neighboring cells. Delta is a ligand and notch is a receptor. On receipt of signal from delta, there's a transduction process, which in this case, actually, the notch protein itself is cleaved, and the intracellular part goes off and signals to the nucleus to change gene expression. And there's a whole bunch of responsive genes to notch signaling because it regulates very, very diverse sets of fate decisions in cells. But the basic idea is that the level of notch signaling in a cell controls the fate. The way it does it, again, there's a lot of detail known to this that isn't in this picture, but it boils down to this, that you have downstream, you have a set of genes which, depending on the organism you're in, are called Hess genes or Her genes or enhancer of split genes. They all have different names, but they all have basically the same function. They can repress their own transcription, but they can also repress the expression of this set of genes called proneural genes. And those are the ones that control fate, the proneurals. So if you have high proneural, you'll go off and be a bristle or a hair cell or whatever the differentiated cell fate is. And if you don't, then you carry on proliferating and you don't differentiate. So this is really the key key determinant of whether you're going to make the decision to differentiate into a particular cell type or not. It's proneural gene expression. They can sometimes activate their own expression to form potentially a switch in their own right. Once they're turned on, they stay turned on. But they also activate the expression of delta, which then closes a loop in here, because now increased notch activity in this cell will increase the Hess type, decrease the proneural, and so decrease the delta. So there's actually if you follow the linear logic through here, notch activity represses delta activity in the same cell. And so that's the basic logic of what's going on in here. And what's interesting about this from a modeling perspective is that we have a set of interlocked feedback loops in here now. We've got a couple of cell-autonomous loops. There's a cell-autonomous negative feedback loop in here. There's quite often cell-autonomous positive feedback loops. And then we've got the overall intercellular loop where delta signaling will tend to decrease delta signaling in the neighbor. So there's a whole bunch of different interlocked feedback processes going on here. And so the question is, how do all of those different feedbacks interact with each other to regulate the basic function of this circuit, which is cells making robust decisions as to what they're going to do? And I've put these in dashed arrows as well. You can, this is called cis inhibition that I'll mention right towards the end. I'm going to ignore that for most of the talk, which is what most people have done for most of the last 30 years or so, is ignored that. And it's only recently become clear why that's important. Okay, so we've got, this is extremely simplified in terms of the known molecular interactions. It sums up the regulatory logic that we believe to be going on in this circuitry. But what we want to do with the modeling is to try to go into here and work out what's the core logic that's actually controlling the fate decision mechanisms that this network underlies. And also to look at reduced networks to really try to start to pull this apart and find out what the different loops are doing and how do these different loops interact with each other. So that's what we've been trying to do with modeling of this circuitry over the last few years. And the simplest way you can reduce this down goes right back to the original genetic data on these systems. So I showed you the fruit fly example of the bristles, and the main overall logic of this fate decision process was laid down in the late 1980s, early 90s by genetic studies, basically, where you would make clones of cells carrying different gene dosages of delta and notch, and then you'd look very carefully at what happened at the boundary between these different cell types and how that would affect bristle decision processes at the boundaries. And lots and lots of genetic data were boiled down into this type of picture, that notch activity in a cell would repress delta activity. And that would give you this essentially a toggle switch that operated between neighboring cells, because we have a double negative feedback loop, which is a positive feedback loop, which allows these two cells potentially to adopt different levels of expression of delta and notch through an amplification process based on this positive feedback loop. So that was what came from the genetics. So if we look at that mathematically, then it's very easy to start writing down models of these types of things. If you just really model the genetics, which is this was the picture that came from the genetics, then all we need really is to have, for each of these variables per cell, you need a production term and a degradation term of some type, because these cells you need production and degradation so these cells can be responsive in either direction. And then, I mean, this is the simplest model you can write down. You just have a linear degradation for each of these things and then some basic production terms. Delta signaling, so if I have delta on this cell, it increases the notch activity on this cell, so my F is some increasing function. This just means delta on my neighboring cells in a population. And then G is just some decreasing function to represent this. That's all you need to model this. You don't need any more details. And if you do that, then it's very easy to show this behaves as the kind of switch you'd expect. And if you look at the steady states that you achieve in different types of populations, you get this on-off type pattern. So in a ring of cells, you have low notch activity, which is the cell type that will go on to differentiate low notch, it's not inhibited. In these isolated cells surrounded or interspersed with one or two cells that can carry on proliferating. And in 2 dimensions, you get something that's more or less similar to the pattern that we see in something like an inner ear epithelium in this development. You can equally put in all of those details about the transcriptional logic downstream of this, and you can, again, show that the more detailed models will support this type of behavior, which shouldn't be surprising, given that they're following the same core logic in here. And there are big differential equation models of the whole circuitry that don't really say much more than this in some ways. So that's fine. This is a very rapid decision type process. What actually happens with the dynamics, if we look at a row of cells in here, for example, a ring of cells like we just saw the final state of, then what actually happens is that initially, at the start time, you can have just very, very small stochastic differences between cells, extremely small differences, and they will pretty rapidly sort out into a coherent pattern of notch and delta activity, with each cell definitely having, say, high notch activity and low delta activity, or vice versa, in a nice patterned way. And the dynamics of that are well known. They're just the dynamics of a toggle switch. If you look at this in a phase portrait, looking at the levels of notch in 2 neighboring cells, then if we start off with both cells, we imagine they both had low notch, then they move. We want the cells to be initially essentially equivalent. We don't want them to have different levels of expression. So we'd expect cells to be starting off on this diagonal with roughly equal values of notch. Then they will very quickly move along to this intermediate state, which is actually an unstable steady state. And then they diverge either to a state with high notch in cell 2 and low notch in cell 1, or vice versa down here. It's very classic behavior illustrated by this phase portrait. And the one thing I just want to note from this for later on is that very classic behavior in this type of system, well known from this type of dynamical system, is that these are equally spaced time points in the trajectory of this system. And you see you move very rapidly towards this point, and then you hang around here for a long time, making the decision. So the cells both come to this intermediate steady state, and then they hang around there for quite a while before they decide what they're going to do. Then they shoot off to their steady state up here. If we look at the time courses of that, both cells start with high notch up here, for example, then they come down to this should say N1 and N2, my spacing's gone a bit funny. They come down to this intermediate state, and they hang around there before then either going low or high. And that becomes important later on, that state there where you're hanging around in the uncommitted, indecisive state. This is where the decision is being made. It's there. And exactly who will come out low and who comes out high will depend on very small differences initially. But they get swamped out first, and then they get re-amplified as you go along. So it's very simple. What's happening? You're going from an uncommitted state where they're equivalent to this flip-flop type logic where one of the cells, or a subset of the cells in the population, will adopt a state where they have low notch signaling. That's the primary fate that will go off and differentiate. And the remainder of the cells will stay in a secondary state with high notch activity and will carry on proliferating in general and not differentiate. So this little toggle switch starts off with equivalence and ends up with a stable one-way conversation. This type of cell, the primary fate cell, is signaling in a directional fashion to its neighbors, inhibiting them from adopting the same fate as it, which is why this is called lateral inhibition. These cells, once they've started to go towards the primary fate, will inhibit their neighbors by signaling. And so there are situations where this has become hardwired so that the cells that are going to be primary fate are actually pre-selected by some other mechanism and will just inhibit neighbors, which is why, kind of why this originally became called lateral inhibition. I prefer to think of it as mutual support because you actually need both of the cell types to maintain the stable steady state in here is only maintained when you have neighboring cells of different fates. If you take one of those away, the situation becomes unstable again, and it will reassign a primary fate cell to take the place of this one. So I don't like to think of it as lateral inhibition. It's actually more of a mutual support thing, but it's really just semantics, I guess. Okay. That's pretty much what we think happens in these situations where you need to make decisions very quickly. But I alluded to the fact that there are other situations where you want to be rather more measured about the way that you make cell decisions, and a really nice example of this is in the developing nervous system in vertebrates. And in vertebrates, you're developing inside the mother. You have plenty of time to do it. It's a nice controlled environment. You can take your time over it, do things more leisurely, at a more leisurely pace. And one of the situations where you want to do that is in the developing nervous system. The reason being that, so the nervous system in vertebrates like yourself forms from a structure called the neural tube in the embryo, which runs along your body axis. And within here, this is actually a single layer of cells still. So if I cut this open and laid it out, it's actually just a monolayer of cells that are the cells that will go on to form your nervous system. And so if we looked at a section of this, it's a single layer of cells, but it's a rather strange one. What those cells are doing, this is the cell body with the nucleus in it, and it's a rather thick sheet of cells for a single layer of cells because they have these long projections to the two surfaces. So this is the apical surface here of the epithelium, and this is the basal surface of the epithelium. So this is up here, and this surface is down here. And what the cells do, as they're going through the cell cycle, so the division process in these cells, because these cells are proliferating to generate all the cells of the nervous system during embryonic development. As they go through that cycle, the cell bodies actually migrate up and down. So the cell bodies are migrating up and down in here during the cell cycle. It's called interkinetic nuclear migration. And so the cells divide down at the apical surface in mitosis when they're down here. And then after mitosis, the daughters move up through the G1 and S phases of the cell cycle, and then they come down back to the apical surface to undergo division again. And this takes several hours to actually go through this whole movement. And it's important that they do this movement, it seems. Nobody's given me a really good answer as to why you have to do this. But if you stop it, you get premature differentiation. Because the important thing about forming the nervous system is that you want many different types of neurons. We'd be fairly dull if we only had one type of neuron, like the bristles on the back of a fly. We wouldn't have a very sophisticated brain. And the way that you generate the diversity of the cell types in the nervous system is by doing it over a significant period of time, number of days during human development. You're actually generating the different types of neurons and associated cells, and it's done sequentially. So you have some early differentiating neuronal types, and then intermediate ones and later ones, and it's done in a very measured fashion. And there's all sorts of sophisticated feedbacks that make sure you get the right proportions of different cell types so that you have a well-balanced brain. And it doesn't always work, I know, but sometimes you get mathematicians or computer scientists, but normally it works. You get normal brains. But the point is, you need to do, the cells need to be doing decisions. You need to single out subsets of cells to be neural cell types, but you need to do it over a very slow period. You don't want that rapid decision making that we saw with the simple delta notch circuitry. And there's all sorts of movement going on. So what's going on here? Because the interesting point is that so the decision making in this type of situation is still done by that same network, the notch signaling pathway. You find this in all invertebrates and vertebrates. It's always there, and it seems to have the same basic logic that we just saw. So we've now got the same molecular machinery working, but the time scale of this process is very different. And we have much more proliferation going on here and only a very slow decision-making process. You just occasionally make decisions. So how do you stop these cells making decisions? That's a really key question in here. And again, you can show that the notch pathway is central in controlling the decisions because you can, if you block the notch pathway, then in the case of a fly, that just means no cells get inhibited, and so everything becomes bristle type, and you have no skin, which is not good. You just have bristles. And in the case of the nervous system, everything just differentiates into early type neurons, and you have no later type neurons if you disrupt notch signaling. So the notch signaling has to be there to make this process slow. It also has to be there to make the decisions. So how do you actually square those two things? One possible explanation is that the notch pathway is actually operating in different ways at different times in a cell's life. So here's the neural tube looking at it from above along the body axis with the cells in it. So this is a kind of surface view of it. The one possibility is that you've just got slow transitions between different modes of behavior at that network. So maybe early on, the notch pathway is necessary for actually maintaining cells in an uncommitted state. And then it goes through a lateral inhibition type phase, but still not a really strong lateral inhibition. And then maybe later on, you go to a really strong lateral inhibition where differentiated cells inhibit their neighbors. So it's kind of like slow transitions between different modes of behavior of a network, which is possible, but I'm going to argue that it's not like that. And the reason I'm going to argue that is because of these data. So I showed you the schematic of the notch pathway. We had delta and notch, and then the first downstream components were the Hess type things, the H's, and then the next ones were proneurals. Now, recent technology allows you to actually visualize in single cells the dynamics of expression of these proteins. And so these are data from a group in Kyoto, Ryo Kageyama's group, who's very interested in these processes in the nervous system, in vertebrates. And here you're visualizing, in this case, you're visualizing Hess1, which is one of those H's, one of the immediate downstream targets of notch. And these cells here are precursors, neural precursors in culture. And they have this luminescent reporter for Hess1. You can do the same experiments for neurogenin-2, which is a proneural gene, so one of the P's, and you can do it for delta-like 1, which is one of the delta-type proteins in here. So these are all notch pathways. And if you actually look at the dynamics of these in culture, then the level of notch pathway activity is actually oscillating on and off in these cells. And these are cells that are actively undergoing that decision-making process. And yet what they're doing is oscillating. And the period of this is about 3 hours of these oscillations. And they carry on oscillating for quite a long time in culture conditions. And if you do the quantification of that, the overall level of luminescence, then looking at single cultured cells like this, you see a very clear oscillation in the level of luminescence, which is the reporter of transcription, essentially, of Hess1. And if you look at whole slices from developing brains, from a developing telencephalon in the forebrain, you see the whole population is actually undergoing, it's clearly not synchronous, that the whole thing is oscillating in a synchronous fashion, but you can still see the signature in the whole slice of brain tissue at this oscillation in expression. And so underlying this decision-making process, you've actually got oscillatory dynamics, which was totally unexpected in terms of dynamical behavior of this system. So where's this coming from? To answer that, we kind of, it requires a bit of a backtrack to before those data were actually observed in the nervous system. And a few years earlier, the same group, Ryo Kageyama's group, published these data from fibroblasts, which are actually a range of different mouse cell types that they were culturing and looking at Hess1. And here they're looking at a population level. So these are just samples from a population rather than absolute time courses of individual cells. This was before they had that technology. And this is Hess1 again. And if you look in these fibroblasts and you synchronize them at time 0 by treating them with serum, you see these very clear oscillations. In this case, of a 2-hour period, it's slightly different, but it's a very similar type period. And so the nice thing about this is that there's a very good data set, or comparatively good data set, I should say. It'd be nice to have more data, but it's enough data, both on the oscillations and on things like the half-lives of the messenger RNA and the protein involved in this. And to cut a long story short about that network, it became clear from modeling work that we needed to really worry about how we model production rates in genetic networks. So normally we just happily write down either ordinary differential equations or stochastic models where the production terms are essentially instantaneous. You say, I have a regulator of a process which is regulating the rate at which it happens, and the output appears immediately. There's no time delay in the process. It's just an instantaneous production process. The problem is with transcription, where the reading of the DNA to make a messenger RNA molecule is that you control the rate of that process by the binding of proteins called transcription factors onto the DNA. That's the rate of initiation of this whole machinery, where you have a polymerase that trundles along DNA at about 1 to 2,000 bases a minute. You make nascent messenger RNAs that require splicing because they have sequences in them that don't actually code for the expressed protein. These are the introns, designated in blue here. They get spliced out, you have processing of the messenger RNA, and finally you have export to the cytoplasm. And so considered, as we normally do, as an input-output process where the input is transcription factor concentration and the output is mature messenger RNA that goes out and does the next step of the process, being translated into protein, then this is actually quite a long gestational process. It's certainly not instantaneous by any stretch of the imagination. For a typical fairly small transcription factor protein like HES1, this whole process from input to output probably takes about 10 to 20 minutes. And there are data that support that over quite a wide range of different transcriptional events like this. Obviously, it depends on how long the genomic sequence of DNA is that you're transcribing. The longer the DNA, the longer it's going to take to make a messenger RNA off it. And you can get to rather extreme situations with genes like dystrophin in human, where this process takes about 16 hours and has been studied in detail. There you can actually watch these things growing over time. And this whole thing, from changing the rate at which you initiate this process to seeing changes in output, is about 16 hours in that case. So it really matters. In the case of HES1, so we did modeling work to show that this was important in the HES1 circuitry. That was then confirmed if you actually look at different probes for HES1 that probe these nascent messenger RNAs just growing here, using probes for the intronic sequences that get cut out, compared to a probe for the final output. Then here's the intronic probe in yellow. You see the peak following stimulation at time 0, after about, about 35 minutes or so, and then about 15 minutes later the peak in the actual final output. So there's a visualization of the actual delay between the initiation of the process here and the output of the process down there. So I don't want to, there's a whole bunch of work behind that I don't want to talk about today. The bottom line is that, remember in the top right of my little notch circuitry, there was the H and it had a little negative feedback on itself. And the nice thing about these fibroblast data is that they isolate that circuitry. They're not doing all of the big notch pathway, but they've got HES in there and it's doing its own little negative feedback. And what we were able to show was that rather than using ordinary differential equations, you actually need to consider the time delays both in this transcription process and the translation process that comes next of reading the RNA sequence into protein as well. And that you need to use delay equations of some type. So here's delay differential equations. You can do delay stochastic simulations. The point is you're considering the delays explicitly in here. And then you get, you can reasonably well fit the type of data that we obtained with those, either with a deterministic type framework or with a stochastic type framework in there. What that really tells us is that when we go back, so we were just looking at that, but when we go back to this whole circuitry, we should really be, have I spoken for an hour already? Yeah. Oh, we started, sorry, I thought we started at o'clock. I just looked at my watch. Did I have an absence or something? Sorry. So we've just seen that. That was scary. That was really scary. Sorry about that. Okay, so we've just looked at this part here and shown that we needed to take into account the delays inherent in this to be able to explain the dynamics of just this little bit of circuitry. Now, so taking that into account, we say, well, in that case, we should be considering the delays in all of this, because we know that one's important in controlling dynamics. It generates oscillations in these cells that we hadn't been able to see before, because you need to be able to synchronize the cells in a population to actually visualize these oscillations. So what happens if all of this has got time delays in? Then what? Now we've got interlocked feedback loops with time delays. And mathematically, that means anything could happen, basically. So we need to know what's going on between all of these different loops. So the way that we went about that, rather than just throwing down a whole set of equations, putting delays in and simulating and seeing what happens, because there are lots of unknown parameters in there, we decomposed this into its basic elements. So one way of doing that is to actually say, well, we're interested, we see these oscillatory dynamics in decision-making processes. So let's start with the bit that we know can oscillate. Here are just some Hess oscillators in two neighboring cells with the delay that we need for Hess oscillations. And then remember what the logic of the notch signaling was doing. It was just like each cell is repressing its neighbor. It's that double negative feedback. So what if it was like this? What if we had Hess oscillators and they were just coupled through notch signaling to couple their dynamics? And these would have significant delays in them because they involve several transcriptional steps and signaling steps. And so these would be quite big delays you'd expect in this type of signaling process. And what we find is that they behave like coupled oscillators. So there's been an enormous body of work done on all sorts of different coupled oscillators, and these don't really behave any differently to coupled oscillators in any other setting. What you find is that if you can tune any of the parameters which are governing the coupling between neighboring cells. Here we're tuning the signaling delay, and what you do, you go through phases of in-phase oscillation and out-of-phase oscillation, basically, as you change the signaling delay. You can do this by changing signaling strength or any of the other parameters in there. And if you look at two cells, you're basically going from out-of-phase to in-phase to out-of-phase to in-phase. So this is just looking at the lag between two neighboring cells. So here they're in-phase, out-of-phase, in-phase. So kind of what coupled oscillators do, basically. They synchronize with each other, and they tend to be in phase or out of phase. There's a little bit of a modulation of the period shown here, but nothing very significant. Again, all of that works with stochastic simulations too. It's obviously a little bit messier, but you get exactly the same transitions between in phase and out of phase. So the stochastics don't really make any difference to the overall behavior of these systems. So that's good. It's nice. This is just looking at two cells. If we look at populations of cells, oops, went twice, then we can get all sorts of nice patterns of synchrony and coupling of phases between these things. We can get kind of propagating defects in lines of cells. We can get more interesting oscillations which are not straight sinusoidal looking, where you have kind of peaks like this. You can even have situations where half the cells oscillate and half of them don't. And all of these you can understand in terms of the basic mathematical modes of this system. When we put this into 2 dimensions, so here's a checkerboard. You can do it with irregular cells as well, or hexagonal cells, whatever you like. It's just more of a mess to do it. And what they do, this is purely deterministic, and there were no patterned differences on here, just slight differences in initial state, is that the notch activity kind of comes on and off in this rather kind of irregular way that's different every time you run these types of things, which is good. We get Hess oscillations. This is just mapping out all of those cells in the grid, and so that you can watch each individual cell over time. So they're doing more or less odd oscillations in there. But the problem is they just do this forever. There's nothing to actually make those cells make a decision in here. It's just coupled oscillators. So we can get nice, all sorts of bizarre patterns coming up. Even with this, you get a population average that's oscillating like those telencephalon slices, but the cells never make any decisions. So it's not really what we want. It's nice, but an interesting maths, but really it's not doing what we need it to do. We've lost the decision making. So if you go back to the whole thing, you can do what I didn't want to do, which is throw this at the computer and do simulations. And you can show by tuning parameters that we can either have these sustained oscillations like we've just seen, or we can have very quick decision making, or more interestingly, we can have these situations where cells oscillate for a bit and then do decisions. And that's more interesting, because that's kind of what's going on in the neural tube, is that you have oscillations and then you have decision. So this is what we're interested in. So can we understand where this is coming from by doing a different decomposition of this circuitry? So the obvious thing to go for is go back to that toggle switch logic, but now forget all of this internal dynamics and just collapse it down to what we knew from genetics, that notch activity downregulates delta activity. That's what the genetics told us originally. We know from molecular genetic studies that it's going through all of these transcriptional steps. So let's just say that's a big transcriptional collapse. Collapse it down into one arrow, and it's got a big time delay on it, basically. So we've now got this toggle switch, but with delays. So what do they do? What do delayed toggle switches do? And now we can really start to analyze this mathematically, because it's much simpler than the full network. And what we find is that so long as these cells start off with roughly the same initial state, then they will go through a more or less sustained period of in-phase oscillations, where they're actually in phase with each other. And they slowly diverge apart. And then eventually they stop oscillating and go to their either high notch, or I've put proneural here, either high or low proneural level that determines what type of fate they will have. And so they actually go through a very significant period of oscillation in here. And the period of the oscillations now is actually significantly larger than the delay. So you only need maybe about a half an hour delay to make 2 or 3 hour oscillation period. The oscillation period is always, it's got to be more than twice the delay. And it's, in this case, it's significantly more than twice the delay. You can see here in a line of cells the homogeneity. So these oscillations always occur in this in-phase mode. You can't get the out-of-phase mode here, like you can with the coupled Hess oscillators. This is a uniform oscillation of all the cells in this way. And then, as you see, they start to fall off this oscillation, if you like, and go to these steady states, as we had with the non-delayed system. So this is just the same delta notch pair of equations, but with delays in now. And again, you can do this with the more complete networks, and you get the same type of behavior. So on a phase portrait, what's actually happening now is that these cells go onto this limit cycle. So they're cycling round on here, but it's not quite stable. So if there were never any differences between the cells, it's stable. And they'll just stay on this limit cycle. But any slight difference will still get amplified between them. But it takes quite a long time to do so, because most of the time when they're going round here, the differences shrink. And it's only on small parts of this limit cycle that the differences grow. And so you've got this competition between the limit cycle mode, which homogenizes the population, and the patterning mode, which eventually forces them to these two opposite states of low or high notch. And so the interesting question is, given the particular parameters in the system, how long does it take to fall off here? Now that's an interesting mathematical question that I still haven't completely got to the bottom of, but we've got some insights as to how that's actually controlled by the parameters in here. But I'm not talking about the maths.
Is the scaling similar to your case before, where you had that flat region and then they went, okay,
yeah.
is this longer?
So you'll see that in a minute as to the difference that it makes with the delays. And then this usually doesn't run very well, but let's see. Just a gratuitous movie. Oh, he's doing it every 6 time steps now. It never works properly. You see, it doesn't really work unless I... Maybe I'm in a computer science department. Maybe somebody can explain this to me. If I come out of there and now run it, it shows every time step. But in full screen view, it doesn't. So anyway... So you see, you get these, the oscillations, and then you get the same kind of pattern that you had before without the delays. So it just slows things down, which is good, because that's what we were interested in, is slowing things down. But nonetheless, it's still, you know, everybody's oscillating and then everybody decides. So it's still not quite what we want. But we saw from the 2D simulation of coupled Hess that we had all sorts of heterogeneity coming in because it was 2D. And mathematically, that comes from the interaction of lots of different modes. So maybe if we had all this on 2D, we could get something. Again, you might say, would stochastic kinetics help in here to get the type of heterogeneity that you want? It doesn't. It basically behaves in the same way as the deterministic system. It's a bit noisier, but it still does the switching with these transient oscillations beforehand. And they're actually reasonably tight in terms of the number of cycles that you go through before you do patterning.
Is the delay time random in the stochastic?
In the stochastic ones, yes, the delay time's random. Everything's stochastic. Yeah, so you're drawing from a distribution of delays and all of those kind of things. You can do that with the differential equations as well, of having a distributed delay, and again, it doesn't really make any difference, which is why I haven't shown it. So, I don't know, just a tangential thing. This kind of surprised me when I saw it, and it shouldn't on reflection, because we're quite familiar with this type of thing happening with decision processes. Two which I can think of are when you're trying to pass somebody in a corridor. And normally, you want to do a lateral inhibition process. One of you wants to go to the left-hand side and the other to the right, and you walk past. But occasionally, we get into this situation where, you know, you both go left, you both go right, you both go left, you both go right. It's a kind of embarrassing situation, and then finally, you work it out. That's exactly what's going on here. I haven't done the experiments, but I think probably if you're both drunk and your circuitry is slower, this is much more likely to happen. So trying to pass somebody in a corridor in the pub near closing time, I'm sure somebody could go and do the research and actually watch and see whether you keep seeing these situations emerging. Another example, I've put lag telephone line here, but actually Skype's probably a better example. You're trying to have a Skype conversation and you've got a laggy line, then you both start talking, and then you both shut up. You can't actually do a conversation because a conversation is lateral inhibition as well, or it should be. Somebody talks and the other one listens. It's one of those stable one-way conversations again, which occasionally flips, hopefully. It's not just a monologue. But the point is that if you've got a lag in there, it's very difficult to actually do the decision process as to who's going to talk, because you're waiting for the gap. And because of the lag, you hear the gap, even though the other person's talking. So we're quite familiar with these things. And in terms of the dynamics that I showed for so this is in answer to your question about the timescales. This would be in a non-delayed system. Remember, this is where the decision's going on, is at this point here. If we do the same thing with a delay in it, then what actually happens is that these phases don't change very much. They're kind of proportional. They're proportionate to the delay. It just slows things down a little bit. But it's this bit here. This is where you get stuck in that transient limit cycle, this quasi-stable limit cycle. So it's this bit that really grows. And I should have put what the delay is on here. And I think the delay is probably 1 in this system. And you see you've gone from a decision making in maybe about 8 time steps to 20 with a 1 unit delay. So it really does slow things down a lot. And it's all because of this transient oscillation in here. I know that looks like out of phase oscillations. It's because this is one cell either coming from a high or a low point. So it's kind of slightly confusing, but they're always in phase. So where do we go from here in actually explaining what's going on in the neural tube? And so here's our neural tube again. And if we look at the cross-section from apical to basal, like I said, we've got this very dynamic situation. All these cells are packed in here, and they're all moving up and down in the epithelium as they're going through the cell cycle. And all of these cells are expressing delta and notch, and all the Hess-type things and proneurals, and they do so from several days before they actually start generating neurons. They have the machinery to do it. So what's going on? And we have the data that show that certainly in certain situations, these cells are undergoing oscillatory notch activity during those early phases before they differentiate. So the way that we chose to try to see how this would actually work out in the full neural tube is to use a different kind of approach called cellular POTS models, which some people may have come across. What you're doing here is that, so you actually represent cells as collections of either pixels if you want to do a 2D simulation, or voxels if you want to do 3D simulations. I really ought to have a slide of this, I suppose, but basically what you do is that you have a simplest way of doing it is having a regular spatial grid, and then an individual cell will just be a set of grid points, a connected set of grid points. So, you know, that might be one cell, say, would be just these grid points here, and then these would be a neighboring cell. And you can write down a Hamiltonian for this system that's made up of various different energy contributions from either interfacial energy between neighboring cells, which allows you to do differential adhesion of cell types. You can have area or volume constraints if you're doing 3D. You can put anything you like on here to give you a Hamiltonian, and then you can do a stochastic model of essentially identity swaps between neighboring pixels, so this cell can invade this cell by this pixel becoming shaded. And so it allows cells to move around, which is what cells do in biological systems. So we actually made a POTS model looking like this, which was very hard and not really very useful, and actually had a Hamiltonian constraint that would make these cell bodies go up and down, which you can do it, but it's not really biology. I think more useful was actually saying, okay, all we really want to know is there any effect of all of this cell movement on the signaling process between these cells? Because we know this movement's important, we know it's there, but we don't quite know what it's doing, and we know that signaling's going on in here, and we know that if we just have the signaling, then everybody goes on to this oscillation and then comes off at the same time, which is not what we want either. So could all of this cell movement be contributing to cells coming off at different times? So the way that we chose to approach that was actually by eventually doing a 2D cellular pop simulation. So there's an underlying spatial grid on here, and you can see each of the cells has got this type of structure of just a number of pixels in there. And so we're looking down on the surface of the neural tube now. So we're looking down like from above here. And so we're just looking at the projections of these cells. So each of these cell bodies is actually going up and down in here, but we're not explicitly modeling that because it doesn't help. And so what the POTS simulations allow you to do is to tune the degree of mobility of these cells. You actually do it by something called temperature in POTS simulations, but it's really just the mobility of the cells, how mobile they are. And so if you had 0 temperature, these cells would just sit in a rigid lattice, like this picture here. And as you increase the temperature, they can start to move around a little bit. And so if you imagine these cells making their way through this very densely packed mass, then the cell bodies are actually snaking their way around. They're not just smoothly going up and down like this, because there's all sorts of other cell bodies that they're having to move past as they go through the cell cycle. And I should also say that not all of the cells in here are synchronous in their cell cycle, so they're all going up and down at different stages of the cycle. They're not all going up and down together, or it wouldn't work. So, unfortunately, for various reasons, I don't have the movies of this, but we can set up these types of simulations, and then in each of these cells we can put the notch signaling network. And so we have the same signaling logic going on between these cells with differential equations as we had before. You actually don't need to put delays in if you have the whole circuitry, because it does the delays itself by having lots of steps. We actually have run this without explicit delays, just because it's easier to call on a standard ordinary differential equation solver than writing a delay differential equation solver in this kind of setting. It's the only reason. And so you choose parameters. So if you look at this in a fixed array, you get these transient oscillations followed by differentiation. And then you allow these cells to move around. You allow them to proliferate. So they're going through this cell cycle. So they will have a certain cell cycle time, and they'll grow, double their size during that time, then they'll divide into 2. So you can do, these are all standard things that you can do in a cellular POTS simulation. So these cells are growing, dividing, and the final thing that you need is that when they have sufficiently low notch signaling for a sufficient proportion of their cell cycle, they will then differentiate because they've been released from the notch inhibition. And when they do that, they actually leave the epithelium, because what happens with these cells is that they actually go out of the epithelium eventually once they've differentiated. So they leave the game, if you like. So they actually leave this population here. And what we found, to our surprise, or to my surprise anyway, I thought this was wrong when I first saw it, but it wasn't, is that as we increase the cell movement, so if we start off with 0 cell movement, we can look at firstly the proportion of cells that come out of that population. What proportion of them turn into neurons? And in the case where there's no cell movement, it's about 0.4. And if you look at the balance between the cells that come out of the population, the differentiated cells that are going to go on to form neurons, and the undetermined cells that stay in there and proliferate, these are essentially the neural stem cells, then we have a slight bias towards proliferating cells, but there's a fairly even balance. And the total number of cells is quite low in here. What happens as we ramp the temperature up, as we allow the cells to move around more, is that we shift the balance between proliferating cells and differentiating cells. So the number of differentiating cells actually stays reasonably constant in here. These are the ones escaping these oscillations and getting low notch for long enough that they can differentiate. But your pool of proliferating uncommitted cells actually grows as you increase the amount of cell movement to a certain limit. The proportion of cells that differentiate over a given time interval actually continues to go down because you're shifting this balance between proliferation and differentiation in here. And so by changing the degree to which these cells are moving relative to each other, you can control the rate at which you differentiate cells from this population and how much of a progenitor pool you leave behind. And so if we were normally, say, up here, this is a very mobile population, if you suddenly block movement in here, you drop down to this type of situation where you actually differentiate more cells. You differentiate a much higher proportion of cells and leave far fewer of them proliferating. And in the case of the nervous system, if you do that early, then that's exactly what happens. You do early differentiation of early neuronal types, and then you no longer have a sufficient proliferating pool to generate later neurons. And so that's exactly what comes out of this Potts model. The other thing you see is that this is looking at notch activity in these cells. It's very heterogeneous, and it's certainly no longer that you have a uniform oscillation in this whole population. You now have cells going on and off, in phase, out of phase, all sorts of relationships in here. And the reason this is, biologically, is that in order to do that decision-making process, you need consistent signaling between neighbors to push them to the two different cell fates. They need a consistency of signaling. And what happens if the cells start moving relative to each other is that you no longer get sufficient time in here to actually make the decision between two neighboring cells. So you start slowly moving apart in this time period, but then by the end of it, you've changed your neighbor, and you've no longer got a consistent signal with your neighbor. And so you end up actually falling back. You kind of hang around this place for a lot longer, and you oscillate while you're there, as we've seen. So what this suggests is that we actually have an interesting balance between the signaling and the movement in here, and that relative cell movements can actually regulate the decision process mediated by this signaling network. And that if you don't have cell movements, you do very rapid decision making, or if you've got delays, you can have delayed decision making because of oscillations, but still people decide at the same time. The decision is made simultaneously. And that you actually need the combination of the movement and the delayed signaling network to actually generate this slow decision process where you throw off cells into differentiation at a nice measured pace. And that pace can be regulated by movement in here. So the movements become very important in regulating the decision process. And finally, so I'm not going to do plants, obviously, but finally, this is, so again, data from Ryo Kageyama's lab looking at other stem cell types. So the neural progenitor is essentially stem cells in there. And so this is a question more generally of decision making by stem cells. And if you look at mouse embryonic stem cells, which again express HES1, but this is by no means necessarily restricted to HES1, it's just that those are the reporters that this group looks at. But if you take these cells and you maintain HES1 at a low level, then you differentiate neural cell types. If you maintain HES1 at a high level, then you differentiate mesodermal cell types. And that if you actually look at them without controlling the HES1 levels oscillate, again, in these stem cells. If you lose the oscillations, once again, you get premature differentiation, and it's much more homogeneous. These stem cell populations, in embryonic stem cells, you need to generate many different cell types. Neural and mesodermal different cell types. And if you block the oscillations, they all differentiate either into neural or mesodermal, which is not what you want. So again, it seems in this case that the oscillatory transients are actually maintaining the stem cells in an uncommitted state, potentially, and regulating the rate at which you differentiate. So I would argue that one of the things underlying stem cell heterogeneity is actually the fact that the stem cell state itself is probably a dynamic state in one of these. It's in the decision-making region of phase space, but it's not just sitting there, it's dynamically moving around it. And it's exploring the different potentialities in a dynamic fashion. And you need to stay on that dynamic attractor to maintain in the stem cell state. And that you throw things off into different states of commitment as you go around this dynamics. And that the actual cell, the details of the environment in which those cells find themselves can regulate the tempo of that process, as we've seen with the POTS model just now.
So is POTS model like a model of magnetic systems?
Yeah, so
it's like an anti-paramagnetic phase transition. Because if everything moves around, it's like a new field.
Yep. Well, yeah, I mean, they're not moving around that much. They're not. They're maybe exploring a couple of neighbours, but not.
But anti-paramagnetic systems have to be quite ordered to reach a stable configuration.
Yeah.
A little bit of disruption.
I don't think they oscillate though, do they? But yeah, I mean, I think there are.
They have a different dynamic.
Yeah, but I mean, the POTS model comes from that physics background, definitely. I'm sure very interesting things could be explored there. So just final slide just to mention, if you go back to the fruit fly, and I said it does the decision very quickly, but it also it has all of that transcriptional logic too. The transcriptional logic I showed at the start is also there in the fruit fly. So why doesn't it oscillate? Why doesn't it sit there oscillating, not making bristles? How do you escape those oscillatory transients? And so this has been known about for a very long time, that you can actually have what are called cis interactions within the membrane of a single cell between delta and notch. It's called cis inhibition. And if you put just a little bit of this in the models, it kills the oscillation straight off, because it basically short circuits all of this circuitry, and you have very quick protein-protein inhibition, which doesn't have any significant delays associated with it at all. So just a little bit of cis inhibition in these models actually kills off. Again, look at the timescales. You're going from 18 to 4, and it's only a teeny little bit. You can tune the strength of this. Just put a little bit in, and it's been shown recently over the last 2 or 3 years in Drosophila that if you don't have these, you don't do decision making. So you've got all the notch circuitry that's meant to do decision making through all this, and actually you need those to actually make it do it. And otherwise, they won't do the decisions. They just hang up, basically. It's not been shown that they oscillate, but I bet they do. So I bet if you take these out, they go into this oscillatory state. And the fact with Drosophila development is that you've only got about 2 hours to do the decision-making for bristles, for example. And if you haven't done it by the end of 2 hours, it moves on. So you lose the chance. So you can't, you know, you get to here, nobody's decided, and then that's it. The window of competence has gone. So you can no longer make a fate decision. And that's exactly what happens if you block cis inhibition. So this was something that was always hanging around in the literature, but never seemed to have a good reason for being there. And then the data have now shown experimentally that you do really need it to do decisions, and this is possibly what's going on, but I can't show you oscillatory dynamics in Drosophila just now, I'm afraid. So just to summarize, we know that, theoretically at least, this directed signaling between delta and notch can underlie very fast and robust lateral inhibition. Those types of toggle switches don't really do anything else but decision making. You don't need any local positive feedback necessarily, but that could be in there as well. But the circuitry, the intercellular signaling, will do it for you. But we saw that it's hard to control the balance between proliferation and differentiation, even where we have the oscillatory transients, because everybody still differentiates at the same time. And then we saw that once we actually take into account the movement of cells, the heterogeneity in the actual signaling, then we find interesting ways of actually regulating the balance between the proliferating pool of cells and the rate at which cells come off and differentiate, which is potentially interesting for a whole range of different stem cell type populations. So I will end there. Thank you for your attention and take any questions if there are any. Mm-hmm.
And do you think that's the main reason for these two different modes? And it's kind of odd that the one that's quick is so complicated.
Right.
So you wonder also if the other one is the default, and then that quick thing is, I don't know.
Is an evolved.
Yeah.
Yeah, quite possibly. And I mean, I would argue that it's evolved to do these cis interactions to actually stop it behaving like a slow decision maker. Yeah.
Over a long period of time, and then suddenly, oh, well, now we need something that.
I mean, the other thing is that this circuitry is also used for doing segmentation. So it's used for the periodic oscillations actually turning into a segmental pattern in insects as well, the same network. So possibly, if that was the. It's always the problem with development is that you're using these networks for very different things at different times. And so you need it to be able to oscillate and to do synchronous oscillations, to do segmentation. And if you want to use it for fate decisions, you fall into this problem of having very long transient oscillations. So you then have to engineer something that stops them, like the local interactions. But yeah, I'd imagine that the ancestral role of this is actually as a kind of oscillation generator, a coordinated oscillation generator. But, I don't know. Yep, can do.
Great. Well, let's thank Nick again. Elaine.
Oh.
Do you want to say anything about next week's?
Really important slide. Sorry, I should have forwarded on. I should have acknowledged Hiroshi Momiji, who is a postdoc here, actually. Sarah Shepherd, a PhD student, and then in the dim and distant past, Sirin Beslingstad and Erik Plater in Norway, who also worked with me on this work. Sorry.